extension specialist with Texas A&M and Texas AgriLife Extension Services. And on today's webinar, we will be talking about managing acidity in red wines, specifically alternatives for acid management in red wines. Today's presenter is Dr. Federico Casassa. He is a full professor of Enology and Sensory Analysis at California Polytechnic, Cal Poly, in San Luis Obispo, California. The focus of Federico's research lies in the broad areas of grape and wine chemistry and biochemistry and their sensory implications, with a special emphasis on applied research. He has been involved in research projects focused on the impact of irrigation techniques, especially regulated deficit irrigation. He is also interested in the chemical and sensory aspects arising from intrinsic variations of berry size in red wine cultivars and their impact during winemaking. Current and ongoing research projects include studies on the extraction of phenolics of sensory relevance during the course of different maceration techniques in red wines, such as cold soak, extended maceration, saignée, co-fermentation, stem and whole cluster additions, coupled with other regulating factors such as grape maturity and composition, ethanol content, SO2 doses, etc. Also interested in hyperoxidation, oxidation reduction potential, acid chemistry, and biochemistry in wine, and their sensory impact as evaluated by formal sensory evaluation techniques. And with that, Federico, thank you so much for being here today. Please take it away.
Well, thank you very much, Andrea. Hopefully everyone can hear me fine and clear. So first and foremost, it's a pleasure to be part of this seminar. I was telling Andrea that I have a deep appreciation for Texas. Have been there 2 times, and I know a little bit about the basic aspect of viticulture and grape growing in Texas. A little bit, enough to cause trouble, I would say. But today. What I'm going to be presenting is kind of an array of alternatives for acid management, especially in red wines, which I know is of particular relevance for Texas. Before I get into that, I would like to provide the audience with an overview of where I am and what we do in general, because we do quite a bit. So we are located in the central coast of California, and this is the central coast of California. Essentially, is a stretch of close to 300 miles, really. And what we have as a defining factor is many valleys that are essentially open to the ocean. They are what I call parallel valleys. We have an array of these valleys that are open to the ocean, and essentially that afford us a very long growing season, a cool growing season, very different from Texas indeed. And this fact, essentially, because we have like a long protracted growing season, allows us in particular to grow Mediterranean varieties or varieties with Mediterranean vocation, and by and large ultra premium and ultra premium fruit. Okay, so I'm just showing you here some of the appellations, and in particular for my research and my research group. So we cover more or less out of this stretch of 300 miles, we cover more or less a range of 185 miles. And in blue, you can see all my research sites. So as you can see, we cover pretty much all the central coast of California, going from Monterey to even Santa Barbara County. Okay. So just the last harvest 2024, because at this point in time I have 2 lab technicians, 5, 6 grad students actually, and 9 undergrads. So we are able to pump a lot of research. These are some of the projects that we completed last harvest. So we crushed, just for research purposes, almost 21 tons of grapes. And I was telling Andrea that before I get into this, I would like to give you kind of a quick overview of what we are after. I'm not going to touch base on the results, but we have been doing quite a bit of work on the effect of hyperoxidation on white wines. So essentially building flavor through hyperoxidation, also controlling and managing NTUs and seeing the effect of different turbidity levels on the final sensory and chemical composition of the wine, again as a tool to build flavor and mouthfeel. We have a model for extraction of phenolics in the context of cap management. We published a kind of a hint of this model already. The full explanation of the model, the full model is a little bit more complex than that. Looks like something like this. So as you can see, effective phenolic extraction in red wine is something relatively complex. And we have a model to actually model and explain how phenolics end up and get effectively retained into wine. As Andrea was mentioning before, this is a very exciting piece of information and research that we are actually producing, which is monitoring the oxidation reduction potential and see if we can actually predict phenolic extraction, which I can tell you we cannot. But what we can actually predict indeed is the late release of hydrogen sulfide or reductive aromas after the wines have been bottled. We have published this. This was published maybe like 2 months ago or something like that. And then we have a model for the potential late release of hydrogen sulfide in wine, in which we believe that the potential for these reduction aromas to show up in your bottle is more of a function of the amount of mixing during cap management than the type of gas that you are used to mix the wine. So it's kind of an interesting results. So just to give you an overview of what we are after, and maybe to pique your interest, and maybe I get invited to go to Texas in person at some point. Okay, so with that being said, kind of a brief introduction, this is what I'm going to kind of touch base today, which I think is going to be relevant, especially if you guys are in Texas. If you are not in Texas and if you are not battling with high pH wines, I have to say we don't battle with high pH wines. We don't have that inherent problem. But what I'm going to showcase is the fact that this is a problem that is more or less transversal to many industries and regions regardless, okay? But if you are in Texas, I think this is going to be of particular relevance for you. So I would like to present you with the scope of the problem, which I think if you guys are mostly winemakers, you should know or have an idea of what this is, and the concept of wine balance as well. I'm going to give you kind of an overview of the fundamentals of acidity and pH, and I'm looking forward to share this with you in the most, I would say, understandable fashion, because this is not easy to understand and it has been traditionally misunderstood, causing really confusion within winemakers. And then I'm going to present some data, and then we are going to discuss the results. So the scope of the problem is clear here. So we have the situation of cyclic climate change as well, the fact that sugar accumulates much faster and acid degrade at the same rate in which sugar is being accumulated. But I always tell my students and sometimes my peers that the fact that we are experiencing this decoupling of sugar accumulation and flavor and acid retention has mostly to do with the fact that we have 60, 70 years of accumulated research on viticulture, in which we have taught and tell grape growers that the bottom line is to optimize photosynthesis, and that come at the expense of acid retention. Okay, so essentially we have been teaching people to grow grapes faster and to accumulate much more sugar and to optimize photosynthesis. Europe typically plant on south facing slopes, much more sun exposure, so on and so forth, optimizing photosynthesis. And maybe, I guess, as a hint of suggestion for you, maybe it's time to change your viticultural practices to kind of slow down photosynthesis in a way. Another aspect is that this is very well known for commercial reasons. We are actually forced to really grow varieties that they don't have Mediterranean vocation. You know, Cabernet Sauvignon, Pinot Noir, Chardonnay. These are varieties that come from cool climates. Some of them, like Bordeaux varieties, from climates that are maritime in nature, not Mediterranean. So these are not, I mean, like they can grow in Mediterranean climates, no problem, but they are not adapted to Mediterranean climates, which is what we have here in the central coast of California. The bottom line is that we end up with wines that are relatively high in pH and low in TA, and that essentially, as you know, compromise the stability of the wine. There is potential for microbial contamination, lactobacillus and pediococcus, and there is the problem of retaining enough free SO2. We know that as well. There is a problem of the wines being oxidatively unstable as well, and compromising aging potential. But what I want to share with you is one important aspect, which is the aspect of wine balance. Yeah, and this is my interpretation of wine balance. I don't know if you are going to find this on the book, so to speak. I don't know if this is formally published, but we do some work on the structural elements of wine balance, and essentially this is the way it can be visualized. You have sweetness and primary fruit in one side of the scale. You have acidity and tannin and tertiary aromas when the wine aged in the other. And then in between you have alcohol, kind of ethanol moving back and forth. But as you can see, I have put the arrow of alcohol more leaning towards the left. Because it does provide sweetness, body, and almost like a sense of primary characteristics. The problem is that we have pushed the scale too way to the left. Yeah, we are showcasing primary fruit, which we like. We are getting increasing ethanol levels, and in order to get the wine back to balance, we really need to focus on acidity here. Tannin and tertiary aromas are not the way to go. Tertiary aromas happen upon aging, and tannin are something that you don't necessarily want on your wine because of issues with potential astringency. So our way to balance, or to get the wine back to balance, is through controlling and managing acidity, which is what I'm going to show you in the next slides. So the approaches are several, okay? And I'm pretty sure that you are familiar with most, if not with all of them. So you can do chemical acidification. These are the three acids that are allowed federally and statewide in the United States: tartaric, malic, and fumaric. Fumaric is an interesting acid to be added, and I'm going to show you why. Then you can do what people call microbial acidification. There are a couple of Saccharomyces species that produce acetic acid. Some of them produce succinic acid. But what is interesting is to touch base on this non-Saccharomyces yeast, which is Lachancea thermotolerans. I'm going to show you some results. Ion exchange, essentially exchanging potassium for protons, or protons effectively, using a resin, which I'm going to touch base. Electrodialysis, which is a little bit controversial in that it actually uses quite a bit of water, and it does produce a brine that has to be treated. So it's a little bit more cumbersome, so to speak. And they are actually indirect approaches to preserve acidity, which, I mean, this sounds like silly and facetious, but you can pick earlier. Sometimes that's actually compromise flavor development, but you can always pick with 20 bricks instead of waiting for 25 bricks and preserve some of the malic and tartaric acid. You can block malolactic fermentation even on red wines. I'm going to be presenting some of that as well. And then last but not least, this is not a solution for everyone, but I can guarantee you that in certain climates, especially in which they are prevailing winds, this is actually a solution. You can wash grapes, and essentially this is what you get when you wash grapes. Most of the potassium is essentially located on the cuticle, and this is the way the water looks like after washing grapes. Yeah. So this particular experiment was done like maybe 2-3 years ago, but what we found that in this washing water, I mean like the water that we were collecting after washing these grapes, this is Graciano, was close to 3 bricks. And out of these 3 bricks soluble solids, mostly was potassium and solids, not so much sugar. So you are removing a lot of potassium just by washing the grapes. Again, might not be a solution, but there is a potential, and there are wineries that have setups that are similar to processing plants for fruits, apples, cherries, that actually are washing grapes today. So I'm not showing you anything new, at least not in my mind anyway. So moving into the fundamentals of pH and acidity, which I think hopefully you value the input here because it's a complicated topic. So I'm going to be distinguishing between pH and TA. Some of you intuitively know the difference between the two, but I'm just going to make it crystal clear, hopefully. So pH, as you know, it measures the concentration of ionizable protons, and it's a logarithmic scale, okay? So it's just the free protons that an acid is able to provide to the solution, in our particular case, in wine. Yes. Now there is a trick in wine, and wine is not milk, and wine is not water. Much more complicated. There is a trick in wine, and the trick is that the concentration of ionizable protons, effectively pH, depend upon the pKa. And what is the pKa? This is a relatively complex concept, but I'm going to make it hopefully simple for everyone, really. So what the pKa is the pH in which the acid exceeds 50% as ionizable and 50% as not ionizable. Again, that's a mouthful, but this is what you need to remember. A high pKa in an acid, let's say acetic acid for fancies, a high pKa means that those protons can be titrated, but they might not contribute to pH. So here you see the titration curve of acetic acid. You can see the pKa, the pH in which 50% acetic acid is in this form and 50% is in the ionized form. And the bottom line, and what I would like you to remember, is what is there is that this acid has a high pKa. That means that those protons are non-ionizable, and a low pKa means that those protons are fully able to contribute to pH. So low and high relative to what? To 3.6, which is the average pH of wine. So remember that if an acid have a pKa above 3.6, that might be titrated, but it's not going to contribute to pH. And any acid with a pKa below 3.6, ideally below 3.4, is going to contribute to pH. Okay, so acetic acid with a pKa of 4.76, it doesn't do anything for pH. It can be titrated, but it doesn't do anything to pH. Now, we know this very well. I'm originally from Mendoza, from a hot region, so it is not allowed for us to add HCl or sulfuric acid. We do it anyway, and I'm assuming some of you might do that as well. If you do, you are going to see large effect in pH, because you are actually adding an acid with a pKa of -7. So that means that if you are adding, which is completely forbidden, it is not allowed, but if you are adding that, you are adding an acid in which all the protons are going to straight and directly contribute to pH. This is not allowed, but this is what happened from a pure chemical standpoint. So again, compare the pKa of -7 of HCl with the pKa of 4.76 of acetic acid, and then you can understand why HCl and sulfuric acid are so effectively in almost breaking the buffer capacity of juice or must or wine. Now I'm here presenting you with all the pKa's of the main acids that we can actually effectively add in wine. So I made very clear that you cannot add HCl or sulfuric acid, but you can add those. And really, what you can add to have any effect on pH is tartaric acid, because of this pKa of the first carboxylic group, and fumaric acid as well, which is 3. Citric, lactic, and malic are not going to do much for you when it comes to moving pH, and as a matter of fact, they probably are going to contribute with counter in a way. Yeah, so they have relatively high pKa, so they can be titrated, but they don't do much for pH. Okay, but tartaric acid does, as you can see, by virtue of this pKa, the first carboxylic function, and fumaric acid, which is an interesting acid, it is allowed. We are going to do some research on it, and I don't know if you guys have any experience, but I have been told that it also kind of provide like a fruity, kind of a strawberry-like character. So we are actually keen on taking a look at fumaric acid as well. The pKa tell us that there is at least one carboxylic function that can actually contribute effectively to pH. The rest of the acid will not do anything for you. And as a matter of fact, if you have an imbalance of acid, maybe a little bit more malic acid than tartaric acid, that means that you are going to have a relatively high Ta, but a relatively high pH as well, which is kind of a quagmire in which you don't want to be. Something common in the East Coast of the United States, by the way. So you know this as well. Microbial growth, free SO2, oxidative stability, all depend upon pH. Okay, so pH regulate microbial growth. It regulate oxidation, reduction, reduction reactions as well. Oxidative stability, microbial stability. Pretty much everything that is pure chemistry and biochemistry is regulated by pH. As far as sensory effect of pH, very little really. Wines that have pH below 3, which are rare, maybe some base sparkling wines, they might have this kind of a mineral type of metallic-like character, and wines with pH about 4, particularly on Viognier, they acquire this kind of a oily, soapy, saline type of texture. Other than that, pH does not affect the perception of acidity. What affects the perception of acidity, we know very well, is titratable acidity, the total amount of titratable acids, simply because our saliva, the bicarbonate ions in our saliva, titrate the acid in the same way that sodium hydroxide titrate acids during a normal titration. Yeah. So that's pretty simple. pH regulate pretty much everything, chemistry, biochemistry, microbiologically speaking as well. And TA is sensory effects. So very contrasting effects, really, which we as winemakers have to put together in a way. So this actually brings me to the concept of total acidity, titratable acidity, and something new that I'm going to introduce to you. New, which I chuckled really, it's not new. It was published in 1982. So the concept is not really new, but it's handy to understand what is going on in juices and wine. So first and foremost, I call you to be mindful when you talk with someone and differentiate between total acidity and titratable acidity. So total acidity is the total amount of acids both in dissociated and undissociated form. Tartaric, malic, and citric, that's what we have in grapes and wines. Okay, so essentially this is what total acidity is. You can measure this by HPLC. It's the total amount of acid, not necessarily the total amount of titratable acidity. It's the total amount of acids. Tartaric, malic, and citric is what I call the skeleton of the acid. That's what you are actually measuring. Now, titratable acidity are the titratable protons from those acids. So this is an example of a wine with a relatively high TA, but if you happen to have like a hefty amount of potassium, then some of these protons are going to be swapped or replaced by potassium, and then your TA is going to be lower. Your titratable acidity is going to be lower. Your total acidity will not change, but your titratable acidity will be lower. And on the top of that, depending upon the pKa of your mixture, some of these protons might not be ionizable, so they might not even contribute to pH either. Okay, so that's a double whammy, if you will. They might get titrated, but because their pKa is high, they might not contribute to pH. So there is that. Now, what is the concept of the extent of exchange? Something cutting edge, and I'm being facetious here. It's not cutting edge. This was actually published in 1980. So the extent of exchange is really what explains the relationship between pH, TA, in specifically grapes and wines, not in milk, not in water, in grapes and wine. Because in grapes and wine, we have the confounding factor of potassium, sodium, and calcium. Okay? So essentially what the extent of exchange is, as the slide is showing, is essentially the fraction of the total acidity, the fraction of the total acidity, the total acid, that is essentially salified with potassium, mostly potassium, sodium, and to a lesser extent, calcium. It's mostly the proportion of the total acidity that is salified with monovalent cations, specifically potassium. I'm going to show you that if you guys, not that I actually have a relationship with them, not commercial, but there is this lab in California, I'm assuming there is also in Texas, called ETS. If you send your grape samples to ETS, you can calculate this extent of exchange very simply in a way that I'm going to show you in a minute. But it's actually by subtracting and by looking at the difference between the total amount of acid minus the titratable acidity. If that difference is high, you are in trouble, so to speak. Okay, I'm going to show you in a minute practically how that's done. But essentially what we have at this point in time is that wine pH is a function of the extent of exchange. The higher the extent of exchange, the higher the pH. Boulton have established essentially that the extent of exchange in grapes and wines is between 20 to 40%, meaning that up to 40% of those protons might be replaced by potassium. That's bad. Okay, when you are about 30%, that's probably bad. When you are about 35, 40%, that's bad. So that's the extent of exchange. The lower the extent of exchange, the lower your potential pH. Your pH also depends upon the acid concentration, how much specifically, and especially if you have relatively high levels of tartaric acid with a low first pKa, your pH is going to be pretty good, low, that means. And the third factor is the strength and proportion of each acid, okay? The main contributor to both titratable acidity and pH in a good way is by far tartaric acid. Okay, but if you have an imbalance, meaning like more malic acid than tartaric acid, then your wine pH is going to reflect that by essentially showing you an increase. Now, in terms of approaches to adjusting acidity, I mentioned the use of Lachancea thermotolerant. I am not related with any of the companies that provide the yeast. We have done research with different strains, and I'm going to show you some of that. What kind of blows me away is why people is not using this more. That's the way I can put it, the simplest possible way, really. I don't know why people are not using this yeast as a tool, because it's really, really a tool. What it does is a non-Saccharomyces yeast that essentially diverts or shunts a portion of the pyruvate. Pyruvate is essentially the main product of glycolysis into lactate. Now, when people see that microbiologists with a little bit of training see that there is a portion of pyruvate being shunted into lactic acid production, they get excited because they think that you are going to get less alcohol levels, and the amount of pyruvate in a molar basis is relatively low, really. So you are not going to see a decrease in the ethanol content, but you are going to see potentially a hefty increase in the lactic acid content upon the use of Lachancea thermotolerant. Another thing that we have found, like very clearly, is that different strains of Lachancea produce contrastingly different amounts of lactic acid. So I'm just showing you two of the strains that we have been working with, and I'm going to present you some results. So there is Lactea, which is from Lallemand, I believe, that tend to produce relatively low amounts. And there is another one called Omega Simaflor, and this is from Lafort, and these tend to produce much higher levels of lactic acid. Okay, so there is large variation within strains A and B. I can share this in the discussion, but generally speaking, the warmer you ferment, the higher the amount of lactic acid that Lachancea tend to produce. So there is another, I would say, point of control for you there in terms of the amount of lactic acid that you might want on your finished wine. Now, another approach that we actually follow is to use ion exchange, which again, properly done, is a fantastic solution. I just don't understand why people don't use it more broadly. I guess it has to do with lack of understanding. And I have been told that in Texas there has been a couple of kind of a homemade ion exchange columns. That's probably problematic, really. But the ones that are professionally manufactured work really well. And essentially what ion exchange is going to do for you, or for us if you will, when we have conditions of high potassium, some of these potassium ions are going to be swapped by protons. Now, from where these protons come from, they come from the column, they come from the resin. This is approved by the OIB. It is approved here in the United States. And essentially what it is a column. This is our model version of this column. It's a very small column. It's called Frica. That's the commercial name. And essentially what it is very simple, really. It's a resin. It's a divinyl benzene resin in which each essentially of the branches of this resin are essentially hanging or holding protons. Those protons come from an activation step that can be done with sulfuric acid or HCl. So you need a level of training, formal training, in order to manipulate the column, because it needs to be activated with sulfuric acid or HCl. But once the beds and the branches of the divinyl benzene resin are essentially activated with protons, you flow wine through, and essentially what happens is that the potassium from the wine gets into the arms, and then the proton is released back into the wine, resulting in a lowering in pH, and not just potassium, but all the cations are essentially gone pretty much. Okay, and then you can treat a portion and blend back, which is what we do. So this is the lab scale model that I requested to be built for me for my purposes, but the columns can be pretty high. Okay, they can literally treat 200 300,000 gallons in one go. My column is much smaller, as you can see, pack much less resin, but the principle is pretty much the same. Okay, so what I'm going to show you now, and looks like I have plenty of time, so I think I can slow down a bit, is some experiments that we have done on at least two varieties that are problematic for us, and one that is not problematic, but we did it for fancies, A, and B, because the variety is kind of unique and it's kind of a hard variety to adjust and manage acidity, which is Nebbiolo. So we have Nebbiolo, Tanat, and Tempranillo. This is one of my grad students' work. So she actually made the wines. I got the fruit. And essentially what we did on the three varieties, the same protocol. So essentially we have a control in which is a normal traditional fermentation. We can go over the basics, but there is not much to add there. It's a regular fermentation. Then we have a treatment in which on purpose we block malolactic fermentation. That means that after the completion of alcoholic fermentation, we adjust the SO2 to 0.6 molecular, effectively blocking malolactic fermentation. I know that the technique works. It's kind of a little bit problematic because after that, before someone asked me, essentially at bottling the wine have to be sterile filtered because you are preserving the whole, I would say, content and amount of malic acid. So that wine is actually unstable. So it has to be sterile filtered before bottling. Then we did fermentations with Lachancea, and then we treat a portion of the wine with ion exchange, and we blend it back at a proportion of 30%. So we are added 30% of the volume of that wine with wine that was treated with ion exchange. Generally speaking, depending upon the starting pH, I'm going to show you in a minute, but we usually start with wines with pH 3.9 or 4, and after ion exchange we make a base wine that is usually pH 2 or 2.1, okay, and relatively high TA, and then we blend that back. We did the same for Tanat, and we did the same for Tempranillo. Okay, so pretty simple experimental design, really. And we produce the wines, and now the wines are so popular, we have been putting the wines in events and so on and so forth. I'm going to send the wines to Spain as well, and speak in Spain as well, that we barely have more wine left, really. Yeah, these are interesting wines to taste, but sadly they are almost gone at this point in time. So in any case, so for the Nebbiolo, I would like to share you results on the Nebbiolo and on the Tempranillo, two contrastingly different varieties, but I think that you guys are going to get the gist. So essentially for the Nebbiolo, so this is fruit from Santa Barbara County. It came with 23.6 Greeks. You know, the TA was 7.6, pretty high, really. pH 3.26, no problem there. Malic acid, regular levels, and quite a bit of tartaric acid. So I was making reference that if you send your samples to ETS, you are going to get tartaric acid and malic acid in grams per liter. If you add them up, that's actually your total acidity. This is your titratable acidity. You can calculate the delta, the difference, and then the proportion of the delta relative to the total acidity gives you an idea of the extent of exchange. So the extent of exchange of this fruit is 27%, pretty low, really, so no problem there. So it's a low extent of exchange. You can also see that, you know, moderate to low levels of potassium, nothing out of the ordinary. This is not problematic fruit, okay? So let me just state that clearly. We did our experiment anyway. So from top to bottom, I'm showing you the results on the control, on the wine in which we block it, malolactic fermentation, the Lachancea, and the ion exchange. And just briefly, what you see highlighted on yellow is when we see higher than average values, and when you see highlighted in aquamarine or kind of a light blue or light green, wherever you can see, is when we have lower than average values. So as you can see here, the wine with the ion exchange is the wine that actually lowered the pH the most. That's very substantial. But generally speaking, all the treatments that we use or apply kind of have an effect on a positive effect on pH. Malolactic fermentation, the wine with malolactic fermentation block as well. And then you can see the levels of malic acid and acetic acid. Yeah, a little bit of more BA on the wine fermented with Lachancea, but this is still 0.6, so this is not problematic at all. For the wine with the ion exchange, we blended at 30%, as I mentioned, but the pure wine, the wine after ion exchange, was pH 2.1, TA almost 9, and all the potassium essentially was pretty much removed, and the calcium completely removed. And this is actually important as well. We can touch base why it's important to remove calcium. So moving into the phenolic chemistry of the wine, pretty much good results. as well, if you will. One interesting thing that we have found in and out is that when the way we conduct our fermentations with Lachancea, and I can share freely and openly the protocol that we follow, which is essentially complete the alcoholic fermentation with this non-Saccharomyces yeast, avoiding co-inoculation, so making actually your life easier for you. So essentially what we see is a substantial reduction in tannin, and we have seen this across varieties. So it looks like the cell wall of the yeast is actually able to bind more tannin, and it kind of affords us a reduction in tannin, therefore mellowing down astringency. The effect on color are kind of evident, as you can see, especially for the wine that was malolactic fermentation blocked, even after adjusting the SO2. Okay, so blocking malolactic fermentation really preserves. I will show you more aromas and as well more color. We did above and beyond really in terms of what the amount of data that we have. We follow the redox potential as well. I'm not going to go into that, but we have results on the evolution of the oxidation reduction potential. We did chemistry on the volatile compounds of this wine. What we have found is that Lachancea usually produce less esters, which is interesting because when we do sensory analysis on these wines with Lachancea, what we actually found is that even though they have more esters, they last longer on the palate. They have much more intensity of fruit on the palate. This is time intensity profiles of the wines with the four different treatments, and as you can see, the duration on the palate of the fruit flavor of Lachancea is much higher than the other wines. Ion exchange comes second in terms of fruit on the palate, and that might have to do with the regulation of pH and the fact that by regulating pH, you actually regulate the volatility of certain esters as well. Our lab is pretty advanced. I hope you don't find this bragging, but the reality is that we do relatively advanced chemistry. We have developed a protocol to accelerate aging wines, in which we have to unpool wines, because we do this in the absence of oxygen. So essentially we can predict how a wine is going to evolve over time. The time frame that we use is typically 3 years of aging. We do that in 5 weeks. So within 5 weeks, we know the evolution of a wine potentially after 3 years. And what we found with this particular Nebbiolo is that after accelerating aging, the wines that were malolactic fermentation block retain more polymeric pigment, and they also retain more color. The same was true for the wine with ion exchange. So the color gain from malolactic fermentation blocking and ion exchange was long lasting. So equivalent to 3 years of bottle aging, we still preserve and retain more color with these techniques. Okay, so that's the Nebbiolo. I'm going to spare you from the Tanat, but I'm going to give you kind of an overview of the Tempranillo. Now, the Tempranillo was actually the problem child here, and you will see why. I know that Texas grows Tempranillo, and I think that this is not going to come as a surprise. But the reality is that the fruit came with 23.5 Brix, the TA was 3.8, the pH was 4. You can see already the problem right there. And then I calculate the extent of exchange at 48%. So this is at the upper limit of all the protons that can be replaced by potassium, really. The potassium was 2300 ppm, so pretty high. So this is a good candidate to work with acid adjustment or acid management strategies, yeah? So huge extent of exchange. That 48% is actually telling me that 48% of the acids of the carboxylic group of those acids are essentially swapped or exchanged with potassium. So that's problematic right there, which is why treating the wine with ion exchange works so well. So the control has a pH of 4.17, and the wine treated with ion exchange and blended back at 30%, 3.66. So we really have fixed the wine, true and true. No, there is no other way to put it, really. So very clear results. The results are clear because the fruit is problematic, yeah, especially as it pertains to ion exchange. Here we use a different strain of Lachancea, which is the omega, and look at the amount of lactic acid produced, almost 6 grams of lactic acid. So this wine was actually undrinkable. The acidity, as you can see, was so high that it was undrinkable. It can be blended, it should be blended, but from sensory purposes, we cannot do sensory on this wine because the acidity was so overpowering, really. The pH was 3.71. You might expect more, but remember that really you are actually producing lactic acid, which has a pKa of 3.86. So this is not going to do anything for your pH. What it's going to do is a lot for your TA, but not for your pH. Okay. Now, if you are shooting for acidity and mouthfeel, this is a good treatment for blending purposes. Okay. I repeat, for blending purposes. As far as the phenolic chemistry, we have plenty here. We see again a slight decrease in tannins with Lachancea. We saw even more decreases in tannin in the tannat as well. What else I can say here? More color preserved with the fermentation with Lachancea and with ion exchange. Here is kind of the aspect of the wines. So you can see more or less what happened after treatment. Same results as well in terms of volatile chemistry. We preserve much more esters with ion exchange and by blocking malolactic fermentation, and we see consistently that Lachancea tend to retain more esters, but somehow the wines are fruity still on the palate. So remember that we are measuring esters, but we are not measuring all the volatile composition of the wine. We are just measuring a portion of the volatile composition of the wine. Last but not least, I want to close with this experiment that we run in 2022, so a while ago. Again, using omega. I think I was one of the first adopters of this non-Saccharomyces yeast. Again, Tempranillo. Look at the extent of exchange of this Tempranillo is 48%, so pretty high, good candidate. What we did here, knowing that Lachancea, and particularly this strain, react different depending upon fermentation temperature, we fermented relatively warm. So this is with peaks of 32 Fahrenheit, so excuse me, 32 Celsius. That will be what, maybe like 100 Fahrenheit, 98, something like that. Less than that, 96, 98 Fahrenheit. So quite, it's not one fermentation, but fermentations with a peak of heat, with a pulse of heat. And in that particular case, when we do this, you are going to be surprised to see the levels of lactic acid. We can produce up to 12 grams per liter of lactic acid, and a total acidity, titratable acidity close to 16 grams per liter of final titratable acidity. So obviously, you cannot drink these wines without blending them first, but the idea is to blend them. I recently read a paper on Lachancea in which the experimenters, the researchers were attempting to do sensor analysis on these wines as they are. That is kind of a silly, really. These are blending wines. These wines have to be blended, otherwise they are undrinkable, really. Okay. So just to give you up the last piece of information. So all the way to your right, you see the wine treated with ion exchange. This, and this one, and you see the final amount of cations. And as you can see, ion exchange effectively remove all potassium and all calcium. It is a little bit of potassium less, but as far as copper, iron, and calcium, they are completely wiped out and gone. And after blending, especially on Tempranillo, because it's so high in potassium, you see a decrease in potassium even on the blended wine. Okay? So that's kind of an overview of what we get, effectively making the point again that ion exchange is a good technique to remove cations as well. It's very effective at that. So what I can say, the main takeaway here is that our study showed that all these approaches, blocking malolactic fermentation, using Lachancea, and using ion exchange, effectively allows us to control and manage both titratable acidity and pH, but they really produce better wine. There is no other way to say it. Another thing that we have found is that not all Lachancea strains produce the same amount of lactic acid. They do produce more when they ferment warm, but there is a large variation in the amount of lactic acid. Some of them produce very little, maybe 2-3 grams per liter. Some of them can produce 5-6-7 grams per liter of lactic. So that depends upon what is your goal. Emphasizing also that if you are doing full fermentations with Lachancea, you should blend those wines with the idea of adjusting acidity, really. Instead of adjusting with tartaric acid, you can adjust with a base wine that has been fermented with Lachancea. Okay? And last but not least, this is a good summary for you. This is a Venn diagram that shows you what each technique brings to the wine. All the techniques tend to provide more stable color. You see that in the middle of the Venn diagram. So blocking malolactic fermentation usually give us a little bit more color, more anthocyanins, and obviously more malic acid. That's kind of a, goes without saying, really. We are blocking malolactic fermentation. Ion exchange is very good at decreasing pH and cations, and the use of Lachancea usually decrease tannins. That can be a good thing. More lactic acid, a little bit more acetic acid, and we see a much more important effect of this yeast, particularly on retronasal flavor and specifically fruity flavors. The duration of the wine on the palate with fruity characteristics. So this is kind of a good overview, kind of give you an idea of what each treatment brings. And again, bearing in mind that we work with very different varieties, really. So with that, that's all I have for you today. I don't have social media. I don't have Instagram or Twitter or anything like that. I only have LinkedIn. And when I post things on LinkedIn, usually I try to post things that are valuable, not personal things, because who cares about personal life, really? I don't. So that's all I have for you. And thank you. If there are any questions that I can feel, I will be happy to take them.
Thank you, Federico. Yes, we do have a few questions. From Russell, he was asking about the effect of Lachancea on the wine aromas and flavors, and I think you've already talked about that during the presentation. I gotta say, I had the opportunity to try some of these wines. Now, I don't know if they were blended or not. I don't remember asking that question, but I remember not being a fan of them personally. Now, I don't know how they were made. They were just brought in here, have try these. But if you think about blending and you look at them as a tool, yeah, as a blending tool, it probably makes more sense than just using them as they are produced. Also, I guess my own personal comment after watching this in terms of Lachancea seems to me that it's a tool more of adjusting TA rather than pH. So be aware of that if you're trying to decide what to use based on on what you're working with and what your goal is as you're picking your tools. Jennifer is asking, how do you handle the rapid aging?
Oh, the accelerating aging. Yeah, sure. Jennifer, this is published anyway. So it's kind of a predicament to accomplish what we have. My background is in wine chemistry, really, and this came handy in trying to understand how to do this in the best possible fashion. So what we have found, Jennifer, is that if we have to predict the aging of a wine, especially in normal cellar conditions, we go for 3 years. The confounding factor is oxygen, not temperature, really. So we have to age the wines under conditions of relatively high temperature. That's 38 Celsius for us. I don't know how many Fahrenheit that is, but it's 38 Celsius for 5 weeks. That gives us an equivalent of 3 years of bottle aging. But as you can see, these are ampoules and these are sealed with a torch. And before that, we purge them with argon, so meaning that there is no oxygen. And when we do that, when we essentially accelerate the aging mediated by temperature without the confounding effect of oxygen, we can predict, we can come up with our numbers and predict this kind of a 3 years equivalent of bottle aging. That's pretty much the protocol. It sounds easy. It is not easy. The ampoules are expensive. It requires a technique to ampoule wines properly. Some of them fail, and when they fail, they don't age as they are supposed to age. Yeah, the protocol is published, but it works really well. So to answer the question, 38 Celsius for 5 weeks, that gives us an equivalent of 3 years of bottle aging.
Thank you. And what's the volume of the ampules?
50 mils, 50.
50. Thank you.
Yeah, so now I have two labs, a sensory lab and a chemistry lab, a wine chemistry lab. And with my sensory lab technician, we are trying to find, this is going to take us 1 year, but we are trying to find like a method to do this at larger volumes.
Mhm.
Because the problem with these 50 million pools is that they don't allow us to do sensory analysis.
Yeah, yeah.
And we want to do that as well. We are getting close. So I think that within 1 year, we are going to have a solution.
That was going to be my next question about the sensory analysis, but
Yes.
makes
Yeah.
sense. Makes
Yeah.
sense. Also, Jennifer, who is from Texas, and we in Texas are quite interested in Tanat, and it's becoming a very popular variety here. So she's asking, what were the results with Tanat? If you'd like to share those.
I can share those if I find them. Um, I don't know if I have them here. Let's see. Um, okay, here. Yeah. So this is the Tanat. Um, which I on purpose didn't show because it's a matter of time. So we use this Lachancea called lactea. It produces low amounts of lactic acid. You can actually see not much, 1.58. That's not much really. And these are the results. The fruit came with 29 bricks. We didn't water back. So the extent of exchange was not terrible, 29%. You can see the potassium here. And just to move on, so again, we blended at 30% the ion exchange. The most effective pH was the ion exchange, Jennifer. You can see 3.86 versus 3.54. That's substantial, really. Those are good results at 30%. Thinking that you can even blend at 40 or 50%, we did it at 30% because we consider that kind of an industry standard. What it was very exciting to see is that all the potassium in the ion exchange one was removed, 97 ppm versus the starting 1100 ppm, and all the calcium was completely wiped out. As far as the phenolic chemistry, this is the phenolic chemistry. Look, I was making reference that Lachancea reduced tannins. Look at that. That's a very substantial reduction on tannins. Okay, so the control is 631 and the Lachancea is 500. So reduction in tannin as well, which is consistent with what I say. And what else? This is the color of the wines. This is the Tanat. That's as much as I can share. Those are results. Looks like, let me see what else I have. Oh, I have all the anthocyanins. Looks like the wines are holding fine, even after accelerating aging. Um, yeah, so I don't know if I answered the question.
Thank you. We have a comment and a question from Ravish. Tannins are well-known enzyme inhibitors. So reduced tannins in Lachancea treated wine, will that indirectly enhance the enzyme activity of Lachancea?
Well, tannins are essentially designed by nature to bind with proteins, and enzymes are proteins. If that premise was actually completely true, then the full metabolism of the yeast will be impaired, and that's actually not the case. By the same token, if that premise were completely true from a biochemical standpoint, Saccharomyces cerevisiae will not be able to ferment like a very tannic wine, and it does. So that means that some proteins, some enzymes, effectively, most of them, I'm assuming, are able to dodge the effect of tannin precipitation. So I will not see any problems on that particular regard. Um, the way microbiologists follow and evaluate a fermentation is in their performance, the ability to consume sugar and to produce ethanol, and we didn't find any effects whatsoever on the evolution of sugar consumption. The opposite, actually, is quite effective, really. I have to add to that gentleman or whoever asked is that we do things a little bit different based upon what we learn research wise. And here in the industry, when Lachancea is used, it's actually used with co-inoculation, meaning that the first 5 days are fermented with Lachancea, and then a Saccharomyces cerevisiae yeast is added. Because I try to be as practical as possible and to try to make the life of the winemaker easier and not more complicated. Our protocols just entail adding the Lachancea and allowing the Lachancea to finish alcoholic fermentation. That actually doesn't happen. Lachancea usually tolerate up to 7% alcohol and then after that died off. And then it's probably like a native microbial population that takes over and finish the wine. But I
Um,
don't see any effect on enzymes or anything like that.
Thank you. Sure you've shown this, but what was the alcohol in the Lachancea wines? Speaking of fermentation and alcohol levels.
So you want to see the Tempranillo maybe?
Sure, whatever is easiest. Oh,
Uh,
okay.
yeah, comparable.
Yeah,
Yeah, yeah, comparable. Let's see the Nebbiolo. I mean, like statistically, it's the same. 13.99 versus 13.83, the same. Let's see the Nebbiolo. Uh,
yeah,
uh, zero point 0.2.
yeah.
I mean, it's statistically significant, but
Y-
really, practically speaking, not much. Zero
Yeah.
point 0.2. Again,
That's remarkable. Yeah.
yeah, because again, when microbiologists see the fact that some of the pyruvate is being shunned
Hmm.
into lactic acid, they get excited. But the amount of moles that go into lactic acid on a molar basis is small. So
Hmm.
this is not going to reduce alcohol by 1%. That is not going to happen.
Mm.
Yeah, a
Did,
reduction in alcohol of zero point 0.2 is, from a sensory standpoint and from a practical standpoint, is
yeah,
completely irrelevant.
yeah. Did you run sensory on these wines?
Yes, on the Nebbiolo only because of a matter of cost, and this is kind of an overview of the which I kind of skip on purpose. Really, the most astringent wine was the control, and Nebbiolo is quite an astringent variety, so this is expected. The Lachancea has much more perceived acidity and much more kind of a purple hue and much more color saturation on the ion exchange and malolactic fermentation block. We did that and we coupled that, as I mentioned, with time intensity. And for time intensity, we were looking for duration of fruit flavor on the palate, and we found that the Lachancea was giving us like an extended duration of retronasal fruit flavor on the palate. That's the only sensory that we have on the Nebbiolo.
And the sensory, I was looking at the PCA, if you could go back there.
Mhm.
I'm curious because since I do sensory for everything, this I assume this was a panel. Did you give them these descriptive characteristics to evaluate, or was that something they decided on, or how was
No, they did. No, this is a classic descriptive analysis. They decide the descriptors
Mhm,
based upon the differences that they saw before, obviously knowing the wine.
mhm,
And when it comes to fruit, what they found is that the wines have different amount of fruits on the palate. The wines were lingering differently on the palate. Maybe actually tannins and astringency was a confounding factor because this is Nebbiolo. So because they found differences in fruit, we challenged them to measure the duration of the fruit on the palate, and we did time intensity. So
mhm,
essentially, what this is telling you, this is intensity for the audience, and this is intensity, and this is time in seconds. So at 45 seconds, all the wines have fade out, but at I would say 5 seconds, the wines reach the highest amount of intensity of fruit on the palate, and the Lachancea has, as you can see, quite more.
mhm.
So that's this is classic descriptive analysis coupled with time intensity.
Thank you.
Mhm.
And as a comment, as you know, I'm also focused on acidity and pH management here in Texas. We've run two parallel acidification trials, which are different than yours because we use different acidification methods. But one that was common was the ion exchange. And to your point that there are homemade ion exchange columns here in Texas, we built our own for our lab because we needed something small. We work with smallish volumes, gallons, rather than hundreds or thousands of liters. And I was super impressed by how effective it is, how quick it is, and also the fact that it does not negatively affect the sensory profile of the wines. We did it for both reds and whites. We did whites pre-fermentation, reds post-fermentation. And the ion exchange wines came out on top in terms of sensory rankings and preference. So that was really interesting for me to see. And I was also really impressed with the precision of the method and the speed of the method. So that was something that I haven't worked with that before, so that was impressive to me. And also inhibiting malolactic fermentation is something that we're actively working on. Again, different methods than you used, but makes sense to me. And I'm trying to communicate that here in Texas, that that's something that winemakers should also consider,
So
and
when you say, Andrea, that you are blocking malolactic fermentation by different methods, what do you mean?
yeah, so we in our current experiment we're using fumaric acid, which has
Oh,
been shown to be able
yeah,
to do that.
exactly.
Yeah,
Yeah,
we're using niacin. We're using bacillus, and I feel like I'm missing something. And combinations of these products. So either individually or in combinations of,
very cool.
yeah, to see what's most effective. We're halfway through, so we ran the first one. We're running the second one in a month or two. So
Very cool.
it's really interesting. And also, we've noticed the same thing in terms of color intensity with the controls and the malolactic fermentation inhibition. Obviously, the fumaric acid, as far as we can tell, is the most impactful in terms of color from preliminary results. But it's really, really cool to see. Yeah.
Yeah. So
And
what I was making reference to the ion exchange, which I'm pleased to see that you guys have your own homemade, but I mean you are scientists and you know what you are doing. The reality is that they have a method to be maintained and they have to be
yeah,
activated
yeah,
properly. And
yeah.
someone was actually making reference in Texas that some of the columns were contaminated with Brettanomyces and with things like that. So that's not fun, you know.
Yeah,
That's
no, no, no,
so
absolutely not. No.
yeah, so that has to be said.
Mhm.
And they do have a protocol that has to be followed. But after the protocol is followed for the winemakers in the audience, I'm telling you that to work with economists really, after all the PTE equipment and safety concerns taken care of, is a fantastic technique. And it's so simple. It is very, very
Yes,
simple.
yeah, that's the thing. It's just like, oh, yeah, it works
Yeah.
very quickly and
Yes.
really straightforward. Yeah, and before we wrap up, I just wanted to let everyone know. So, really cool to see people again from all over the world. Of course, we have our Texas audience, but then we have people from Georgia and Michigan, and then Uruguay and Greece and Armenia and India. So thank you all for being here. This recording will be posted, the recording of this webinar will be posted on my Enology YouTube channel, and this is the link here. So check it out. And if there are many, many other recordings there that you can watch. They're available for you, and then you can subscribe to the channel, and then you'll get updated every time I upload something new to that channel. And if there are no further questions, we have some thank yous from the audience. Thank you, Federico, again for
Thank
your
you.
time and expertise. This was really interesting. Thank you all for attending today's webinar, and I will see you all next time. Have a wonderful day. Bye-bye.
Bye bye.
Thank you.